Good afternoon. Yeah, I really appreciate the organizers for inviting me to give a brief overview of what my lab's been doing. And The title is Breaking Resistance Sensitizing Cancers to Immunotherapy. I'm a medical oncologist here. Most of my time is spent in the lab, but I also see liver cancer patients. This is a problem that we're facing in the cancer field. It's a big problem that a lot of us may already recognize here. It's truly impressive the breadth of projects here. So maybe some of you it's not as familiar, but immunotherapy has really become a mainstay for treating cancer patients. It's available to more than half the patients diagnosed with cancer, but we still have to do so much better. If you look at the uh, plot here, most of the cancers fall below the 30% response rate, and that includes liver cancer, pancreatic cancer, very deadly diseases. And so what are some modes of failure? This is just a nice summary, especially if you're not familiar with cancer immunology, using CAR as an analogy for the immune system to go and attack cancer. There could be multiple problems, right? There could be a wheel problem. The immune system doesn't even recognize the cancer in the first place. There could be a brake problem, acceleration problem. The T cells no longer function once they're at the cancer, or T cells don't receive co-stimulation they need to keep going. And then it could be also road conditions, poor road conditions. So there could be other suppressive barriers in the microenvironment like myeloid cells. And so all of these problems, it's a multifaceted problem, it's a complex problem. And if we try to target every barrier individually and try to stack 5, 6, 7 agents onto patients, it's really not feasible. So we need to do better than that and have better strategies. These are a couple of themes that are going on in my lab, and one way is for translation, and this is very historic and typical. We have done amazing things where we start with bench-based discovery, literature, established targets, and then we continue to translate them towards the clinic. And then the reverse translation is starting with patient specimens. So as a medical oncologist and collaborating with lots of clinical trialists, we have access to patient specimens that are treated with immunotherapy. And then from there, we can understand what kind of issues there were with responding tissues or non-responding tissues and have hypotheses that can then be explored in various models. And then we go on in this synergistic cycle. So in this first theme of forward translation, what are some targets that our lab is very interested in? Well, as I mentioned, it's a multi-pronged problem, so we need a multi-pronged solution. So what could be a multi-pronged target? Well, one example, and this is a category, and there are other, of course, targets out there, but a category of protein tyrosine phosphatases. These are negative regulatory phosphatases by and large, and they have multiple functions. They could also improve the activation threshold for T cells, which kill cancers. They can improve their activation. They can also reprogram these barriers, myeloid cells, to become pro-immune and anti-tumor. So one example is PTPN22. This is type 22, phosphatase non-receptor type 22. And it is known to downregulate T cell activation. It's known to downregulate myeloid pro-inflammatory cytokines. So it's a master negative regulator. It's also expressed in only immune cells, so there's this selectivity already in terms of its targeting. And then we have genetically validated biology in people. So in populations of European ancestry, about 10% have a dysfunctional germline variant in this gene. And in these patients, we have higher risk of autoimmune diseases. And then what we have found is they also have lower risk of cancer. And so this two sides of the coin, higher immune system, lower cancer, very interesting biology embedded in this target. We test this in mouse, so we knock it out of the mice, challenge it multiple tumor types. Here you can see that a lot of them have good immune protection against these tumors, including liver cancer tumors. And then when we add PD-1 therapy on top, and this is a single slug of PD-1 therapy, that some of the mice even regress completely. And so there is synergy there as well. So we have enhanced immune response to tumors, But these are immunotherapy sensitive models. So what about in immunotherapy resistant models? And this is the problem that I highlighted earlier. We're dealing with majority of patients not responding. This is a very high bar model, pancreatic cancer model, where it ignores 5-agent therapy. So 2 chemo plus PD-1, CTLA-4, CD40. This pancreatic cancer model ignores it. So very high bar model. You knock out N22 and add PD-1, it's already 40% tumor reduction, and we add a co-stimulator on top, 4-1BB, it provides near complete reduction of tumors. And so very striking response in this very high bar model. We've, of course, done many mechanistic studies to understand why. We find that there's a distinct opportunity in liver cancer, and I think this is a major problem in Korea as well. But it is diagnosed in the US, over 40,000 patients per year. Overall, in the world, it's projected to be third leading cause of cancer deaths. And then, as I mentioned, as a medical oncologist, I see these patients most of the time, and we treat them with immunotherapy. So it is a mainstay treatment for liver cancer, and only 20% respond. So there's a huge opportunity there. There's no FDA-approved drugs for neoadjuvant or adjuvant therapy for HCC. So immunotherapy market, of course, is enormous. So a lot of opportunity here. So one area that I thought would be interesting to engage in potential collaborations is how do we go after these phosphatases? How do we go after PTPN22? It's an interesting target. It is an intracellular protein, so we need to get into the cell first. So of course, there's medicinal chemistry that we can do, and in fact, we've published on the only molecule right now with proven in vivo efficacy. So this is L1. That's a published compound, and we're still working with a team out in Purdue to improve on the medicinal chemistry. But what about ways to bring engineered proteins inside or knock out the protein inside, like in vivo CAR T constructs or antibody constructs that can bring in antisense oligos through receptor-mediated endocytosis, for example. So what kind of targeting platforms we'd love to engage in potential collaborations in this area. And with this medicinal chemistry tool compound, again, nice proof of concept in an immunotherapy resistant model of HCC, this time is liver cancer, where immunotherapy combination doesn't do anything, really nothing significantly at least, and then L1078 provides, this is the compound we have, significant efficacy. So again, this is the area that we think that would be a great opportunity for exploring collaborations through KIAT. So what about the other theme? And just a few quick notes here. This is the reverse translation going from human studies, clinical studies, human samples. And what we often do with these, especially neoadjuvant platform studies, that it's a very good opportunity. And we've done several trials, and it gives you a huge piece of tumor that we can use to study. Of course, we're doing lots of high parameter discovery using these specimens. And then these discoveries lead to hypotheses that we can test in models that are very simplified and controlled. And then we can preclinically test these targets as well in vivo, and then potentially go on to trials, better trials, right? And so this iteration, we are ongoing with these neoadjuvant trials here. You can see pancreatic cancer and liver cancer as part of the GI oncology program at Hopkins. These are a lot of the neoadjuvant, these are most key neoadjuvant trials that we have done recently. So you can take a look. That's a combination of PD-1, LAG-3, some of the investigational immunotherapies out there as well. And then high parameter and omics level profiling. This is now very routine. You've seen some examples of that already from other investigators. But I'm leading the imaging mass cytometry facility at Johns Hopkins. And so we have expertise in this technology. Of course, spatial transcriptomics or single-cell RNA-seq, and also developing very close collaborations. We have that ongoing with bioengineering team or Denny Wards and Pei Shan Wu. I'm not sure if you're familiar with them, but at Hopkins, we're doing collaborations to really build on these omics platforms and create 3D models that we can really tease out the reasons or correlates of response or resistance. We're also developing a new platform to allow massively parallel, high throughput way of screening how the immune co-cultures and mixtures are responding to certain perturbations, and of course, validating further in other models as well. Just a quick example of what we have done in this way. And so if there are any specimens or any other therapeutics that we want to investigate together, this is a sort of a paradigm that we would be using. So here is one case where it's a neoadjuvant cabo, just multi-kinase VEGF-R inhibitor, nivolumab PD-1 inhibitor. Patients go on to surgery. And then again, this is the just an example of the CyTOF facility. So we've imaging mass cytometry and suspension mass cytometry. We have deep expertise in these technologies to profile. These are rather cost-effective, of course, limited to around 50 parameters, but rather cost-effective platform for discovery. In the blood, you can see more enhanced T cells, so effector memory T cells. Here you can just break it down simply. So immunotherapy is working as it should. For imaging, so on the tissue, we can go 40, 50 markers at a time very easily. And of course, there are probably many other options, state-of-the-art options here. But in this case, we've made a tissue microarray from all the patients, put them on one slide, and ran it in a very cost-effective manner, and was able to tease out all the tumor microenvironment of these 12 patients. And you can already make out at a 5,000-foot view, these are different immune cells, green, yellow, and pink. You can see the responders, people who are benefiting from the treatment, have much more immune infiltration as you would expect. We take these images, make them into single cells, and we do quantitative analysis. So a lot of bioinformatics approaches. If there are innovative bioinformatics approaches, of course, that will be very useful. And then quantitatively, the densities of these immune cells are higher in the responders and then non-responders. So this fits very well with what we expect. But then what do we do with the information? This is spatial information now. And so we take all the spatial information and see what are the relationships that really stick out in the responders. The red shows the responders that are enriched in responders, the relationships enriched in responders, and then the blue, the opposite. So you can see there are a lot of these lymphoid to macrophage relationships that are enriched, as well as lymphoid-lymphoid relationships. So not too surprising, but to distill all of that information into identifying which one is the most important. You can see this is a gene impurity statistic from a random forest classification algorithm. And the top ones here, the highest gene impurity statistics, is the most important determinant of the response classification. So from a given B cell or from a given T cell, the most important determinant of response or non-response are these cell type 10 and 11, cell types 10 and 11. And it turns out these are macrophages. And so cell type 10 is closer to these T cells and B cells in the non-responders, whereas cell type 11 is closer in responders. So why does that matter? Well, it gives us mechanism of resistance, right? And at least a very strong hypothesis here. The difference between the two macrophage subtypes here, CT10 versus CT11, CT11 are the ones with high PD-L1. And then CT10 are the ones that are very high with arginase 1. And CD163 is also higher in these. You can make out these visual confirmations, these macrophages, these brown ones that are arginase 1 high, close to these T cells in the non-responder, and then the PD-L1 high macrophages closer to these T cells responder. So there's a lot of deep detail here, but basically what I'm trying to get at is, of course, PD-L1 high macrophages, if that is a mode of resistance in this tumor, we can overcome it because we have nivolumab here. It's PD-1 inhibitor. So that is the therapy in play. And if the mode of resistance there or suppression there is PD-L1, we're overcoming that. But if it's something else, this could be a correlate a surrogate marker, if you will, of some other immunosuppressive mechanism by these macrophages. But these arginase high macrophages are showing that there's this myeloid barrier that cannot be overcome by PD-1 therapy. So it's becoming a very important area. It's already been an active area of research, is to be able to overcome these myeloid barriers. So if there are any collaborative opportunities to look at these therapeutic targets together, that would be really exciting as well. And lastly, another platform, a sort of a technological platform that we have in the lab that can be used in collaboration of any kind. This is a co-culture platform that we have developed. And often we do all these correlates and they are not linked with function. And so what we have developed is this co-culture of immune mixtures from the tumor with cancer and T cells that can really give us a readout of cytotoxicity or tumor cell kill. And so if we then couple it with mass cytometry, we can do single cell profiling of every microwell. There's about thousands of microwells per slide of stochastic mixtures of these immune cells. And we can make out which of the immune mixtures could be the most oppressive in tumor cell killing. And so this is a high throughput, very cost-effective way to be able to do this. Here are just some images of the tumor cell being killed in each of the microwell, and we're being able to profile the cell mixtures within each of the microwell. And this is a survey that we can do to validate further these relationships. And we're able to recapitulate CSF1R, PD-L1. These are all these markers that we know are immunosuppressive, and also this sort of an emerging marker in the MDSC, myeloid-derived suppressor cell world, as another marker of tumor kill suppression. So all that to say, we have these two themes in the cancer immunotherapy field. Love to engage any opportunities, either in any of the examples, either forward translation, reverse translation. We'd love to be able to engage in more opportunities. So with that, thanks for listening. Happy to answer any questions.